And then just let me know whenever you want to be introduced.
You're up.
All right. So I'm happy to introduce Robert Pennington. He did his bachelor's degree in environmental science and fine art concurrency at the University of Oregon, where he also got a graduate certification in sustainable natural resources at Oregon State University.
It's fine.
Before this, he was in rangeland ecology lab in the Northern
That's what
Great
we're gonna
Plains.
have to do. I don't know why it's.
So people need to mute themselves.
Yeah, if you could, everybody could mute themselves.
It'll be in the bottom left-hand corner. Yeah, perfect. Research lab, and then he was in a rangeland ecology lab at Northern Great Plains Research in North Dakota, where he became interested in competition between native and invasive grasses and their effects on individual biomass. So he was encouraged and interested in going to graduate school to do a thesis, where he reached out to Dr. Walid Saidak because of his ecophysiology work and was immediately interested in this project involving Phragmites due to the real world applications and it being an enormous grass. He started in January 2021, and his advisors are Walid Saidak and Dan Larkin. In his free time, he's also an artist, musician, and overruns his living space with houseplants.
Thank you.
Yeah.
Okay. There we go. Okay, so here in Minnesota, we are home to a native subspecies of the common reed, Phragmites australis. We also have an invasive subspecies that is now on the noxious weed list. Efforts are underway across the state to manage, if not eradicate it. However, a barrier to accomplishing this task has been the continued use of the invasive Phragmites in wastewater treatment facilities to remove water from suspended solids, also called dewatering biosolids. The goal of my research is to support the treatment facilities and their transition away from the invasive by identifying either characteristics of a native that would make it suitable for use in these facilities, or even better, identifying a population that we can use. So I'll start by talking about the plant itself, some of our history with it, some characteristics of the invasion, and then I'll talk about wastewater treatment and my experiment in support of their transition. And then I have a few preliminary results. So let's talk about Phragmites. It is a perennial C3 grass species. It is also known as the common reed, very cleverly, and typically spreads clonally using rhizomes, which are basically an underground stem, stolons, which are basically an above ground rhizome. And then it can grow adventitiously from nodes on above ground culms, so the upright stems above ground. But it can also produce viable seeds, which is sort of an important point in the story of Phragmites in Minnesota. There's a few different distinct subspecies. The two that I'm primarily going to talk about, or only going to talk about, is Americanus, which is a native of this continent, and Australis, which is not native to this continent and is invasive. So it's got a cosmopolitan distribution across the globe. You can find it on every continent except for Antarctica so far. Typically does grow clonally. The rhizomes underground can be very extensive. They grow very large, and along every node point on these rhizomes, it can produce new culms, roots, or rhizomes. The above ground biomass is annual and will die back every year, and the underground biomass will persist. As I mentioned, the above ground culms can grow adventitiously, so they respond to layering. And if they fall over or are damaged in some way, that will typically induce them to start growing new shoots along the length of the fallen culms. Personally, I have seen new growth coming from culms that did not appear to be damaged and had not fallen over. So I don't think anybody fully understands that mechanism at this time, but it does like to grow. It does have viable seeds. It was previously thought until fairly recently that seed viability on this species as a whole was very low, 1% or less. And it was actually an applied plant science alum, Dr. Kettering, who was the first to really prove an increased level of seed viability and show that it can spread through seeds. And first, she was able to show that a nutrient-rich environment would cause plants to produce more inflorescences, and on those inflorescences, they would produce more florets. And shortly after that, she was able to show that it was an increase in genetic diversity within patches that directly correlated with an increase in viable seeds. And this has been replicated multiple times, most recently here by Dan Larkin's group. As these patches grow more inflorescences, they have more opportunity to increase their genetic diversity. And as they increase their genetic diversity, they produce more seeds. So as a result, you end up with these very large, very distinctive stands. The above ground culms can be upwards of 15 feet tall. And once you spot them, you will spot them everywhere. But most of their biomass is actually below ground. About 80% of it is below ground, and they grow very extensive root rhizome networks. Here in this photo, this is myself and Mazer researcher Julia Bonin, and we are not standing on saturated substrate. We are just standing on a rhizome root mat that is floating on top of at least another meter of water. So these are very dense networks that can spread upwards of kilometers. So the above ground growth has been used extensively by humans throughout history, including into prehistory. Here in North America, we have fossilized remains of Phragmites as far back as 40,000 years ago. And we have evidence of its use by indigenous peoples to this continent. So on the left are some decorated arrow shafts that were found in the Sinagua cliff dwellings, along with, I believe, a flute, a smoking pipe, some baskets, and some ceremonial objects, all made out of Phragmites. More, even older than that, across the Atlantic Ocean in the Mesopotamian area, we have evidence that some of the earliest boats that were made in human civilization were done, or at least in civilization in that area, were done with the use of Phragmites lashed together with palm date leaves and covered in a tar-like substance. And that's what those two chunks are on the upper right. There's old ancient boat chunks. Below that is a relief showing what are marsh Arabs being pursued by Assyrian soldiers through the marshes of that region. Mesopotamia is in basically central marshes of Iraq. And marsh Arabs are indigenous to this area. And that area is also typically believed to be the biblical Garden of Eden. So the takeaway there is that the biblical Garden of Eden was very likely in a Phragmites marsh. They've persisted there throughout time. As you can see, they still extensively use these reeds and for their as buildings and boats material. Unfortunately, not so fun fact, a few decades ago, Saddam Hussein drained all of the marshes in this region to drive the people out, which worked. But with the fall of his regime, they have returned and reflooded the area and are in the process of restoring it. That's what's on the right. That is a marsh Arab on a boat and a very healthy looking Phragmites patch. More of a fun fact. It still gets used today as a food, as a building material, as traditional medicine. I don't think the food is widely consumed, but you can go out and forage it. It is not palatable to animals, except for goats, which isn't saying much, but it is palatable to people. And I have collected shoots and eaten them myself. They kind of taste like cucumber, and I think they're very good. It gets used in East Asian traditional medicine. This is Lu Gen, which gets turned into a tea. The rhizomes get turned into a tea, which is consumed to remove heat from the body. In Europe, I would not call it a modern building material, but it was used in the construction of homes, especially in thatched reed roofs. And since many of these buildings are still standing today, Phragmites is still used in the construction of these reed roofs. And the average lifespan of these roofs is about 50 years, which is a lot better than my roof. And so this is the map I showed before with its distribution worldwide. But in the early 2000s, there was a researcher, Dr. Saltonstall, who started suspecting that we were seeing a cryptic invasion of a non-native genotype of this species. And so she started doing microsatellite analysis on populations across the world and using herbarium samples, and was able to identify 3 major lineages. The North American lineage, the European lineage, and the East Asian lineage. For simplicity's sake, I'm going to refer to the North American lineage as native and the European lineage as invasive. But within these lineages are multiple haplotypes, and within that are multiple genotypes. The European lineage the genotypes within that lineage have shown a very high degree of phenotypic plasticity, which sort of makes them well suited for becoming weedy and invasive abroad, especially with effects like climate change going on. So the invasion of the European lineage, we'll just say the invasive Phragmites into North America, was likely done with European colonization. And is sort of hypothesized to be from transported across the Atlantic in the ballasts of boats, who would fill their ballasts in marshes on the across the ocean, and then they would sail across and they would dump their ballasts as they were coming into port. Some of the earliest reports of Phragmites appearing are in these developing port locations across the East Coast. But it wasn't really recognized as an invader at the time. And it wasn't really until the 1900s, the early 20th to mid 20th centuries, before we saw it spread across the continent, sort of theorized to be due to the expansion of road systems and railways. So there's not like one smoking gun on how it spread across the continent, but we know that it was fairly recent. Some of these effects of invasion, you can kind of see here. On the left, we have like a native patch of Phragmites, and we have like a gradient of species. And we have some species co-occurring with Phragmites within the Phragmites stand itself. But the invasive stands tend to form very dense monotypic cultures that creates a very, 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 very dense litter layer that inhibits the growth of other plants and greatly reduces biodiversity. There's been some literature on allelopathic effects. In particular, I found one of rhizomes producing gallic acid in great quantities, which reduces the ability of other plants to develop their root systems. Then I found another paper that said they couldn't replicate that at all. And the takeaway there seems to be that it might matter by population. We just don't know yet. Regardless, we still see these monotypic stands all over the place. And we can still see the immediate effects, which is that they produce these really dense litter layers. To make matters worse, we have hybridization. It was first proven in a lab back in 2010. So this is feels very recent. And it wasn't until 2014, Dr. Saltonstall back at it again, was the first to field identify a hybrid population. It was just a few plants, and they were all clones, so they had all reproduced clonally, but it was confirmation. That's what the upper right graph is. It's The little asterisk there between the middle sort of shows, this was again with microsatellite analysis, sort of an even split between the invasive and the native lineages. Managing this species, the invasive in particular, is very costly. It's very time intensive and it's very costly. So these are survey results from 285 land managers that accounts for 81 million hectares. The reason that number is so high is because a lot of these were federal land managers who manage gigantic swaths of land. The takeaway from this study was that neither amount of time nor the amount of money thrown at managing this species was a reliable predictor of eradication as an outcome. But we do know how to manage it, typically with herbicides and lots of herbicides. These are the two herbicides that show the most effect on the species when applied twice annually in the summer and in the fall. And those show the greatest reduction of new shoot growth the following year. The study on the right did a 7-year study of isolated populations of Phragmites, where they used both foliar application of herbicides and they used a cut and drip method where they cut stems open and basically dripped herbicide directly into the rhizome network. And even then, although they did note that over time, the amount of time and effort they had to spend managing these patches was reduced dramatically, in all but, or in just a few cases were they able to eradicate it completely. So it's very difficult to get rid of once it establishes. And we are working diligently in the state of Minnesota to manage the populations that are here. And you can see it's distributed across the state, sort of centered on the metro area. And many of these populations are being treated. Again, treatment does not imply eradication. So it is an ongoing, constant effort to try to reduce the spread of this species in the state. And we also have not just small hybrid populations, but very large hybrid populations that are sort of the focus of ongoing research at this time. So one of the barriers to managing these invasive populations and reducing them has been the ongoing use of Phragmites in these wastewater facilities. So over several years, these facilities were installed across Minnesota in open air beds. And invasive Phragmites was planted in these beds for the purposes of removing water from wastewater. Now, again, when they were put in, it was believed that seed viability was extremely low in this species, 1% or less. And so most of them went into beds like this with concrete barriers, or they went into underground beds that had a very thick clay layer and berms on each side. So it was thought that because they reproduced or they spread clonally, that this would contain their clonal growth. But 10 years later, you recall that a nutrient-rich environment produces more inflorescences, and the more inflorescences over time, you get an increase in genetic diversity. And as you get an increase in genetic diversity, you get production of more viable seeds. So it's not really that surprising that now we see a large number of invasive populations in proximity to these wastewater treatment facilities. So I'm going to talk about what they get used for in wastewater and make a case as to why we should continue using them and not just remove them, or not just remove the reed beds. This is a very simplified diagram of how wastewater treatment plants usually work. Your wastewater is everything coming out of your residential areas and can include industrial effluent. Solids get removed, screened out and sent to a landfill. And then the suspended solids, the basically sludge, moves on through a series of aerobic and anaerobic digesters that removes nitrogen, phosphorus, ammonia, undesirable elements. And then they move on to basically what is a settling tank, and the solids settle out of the mixture, and then the fluid gets hopefully disinfected, typically with UV, and then it goes back into our waterways, while the remaining solids would just go to a landfill. So that's kind of gross. And in the 1950s in Germany, somebody at the Max Planck Institute decided to start trying to use plants to actually dewater the biosolids instead. So they started putting in these plant beds, and they very, very quickly, of course, identified the European lineage of Phragmites as exceptionally well suited for removing water from biosolids. And so this is kind of a cool technology. It's a green technology. You can use these reeds at just about any stage of wastewater treatment. And it has not really taken off in North America, but across the globe, you can find them, especially in developing nations or impoverished communities that don't have the resources to build extensive wastewater treatment infrastructure. And in some cases, reeds are their wastewater treatment. So it's a very, I think it's an important technology to sort of move forward with, especially as we're trying to find ways to reduce our energy expenditure. So here in Minnesota, we have removed our invasive Phragmites from most of these facilities. There's still a few that are using them. It is up to the facility operators themselves whether they want to remove or continue using. But in most cases, these beds are just being used as, or in some cases, these beds are just being used as open air drying beds. And in a couple of different facilities, they have installed native Phragmites to use, but they are reporting sort of mixed results. They're not getting the results they're looking for. And so that is sort of a barrier to getting these other facilities to adopting native Phragmites in their open air beds. And a lot of that has to do with the perception of the native Phragmites versus the invasive Phragmites, which I think, as you can see here, this robust characteristic held by the invasive is not necessarily true for the native. They have less column density, less foliage. They don't grow as tall. They don't grow. Their growing season is a little bit shorter than the invasive. That's the perception, and so that's where my set of experiments comes in. First task was to collect from native populations of Phragmites in Minnesota that appeared robust. And robust was just a classification given to different stands of Phragmites by Mazurk for native stands that had visual characteristics that made them look similar to invasive stands, greater density, more foliage, just more robust growth. In the words of Julia Bonin, it was, if it made her pull over and go check it out to make sure that it wasn't invasive, that's what a robust population is. So I collected from these. Within these populations, I had a series of tracking growth to select candidate individuals that exhibited robust growth, and grew these in a common garden environment to do 2 experiments on, to phenotype their water removal characteristics. Now, because this is sort of a time-consuming process, what we would like to do is identify proxy traits that would indicate high rates of water removal in these populations to allow for rapid identification of these in the field. So As I said, it collected from across the state. You can see the difference here between a robust native and then sort of a weak native. I have a weak native control. I'll explain that here in a little bit. But we wanted to get geographically isolated populations, robust populations. So we tried to get these as spread out as possible. And then the weak native up in Roseau County. And then as a positive control, collected from a population in a wastewater treatment facility that is invasive and was known to be very good at its job of water removal. So the first step after collecting was propagating these things. The literature surrounding Phragmites is extensive, but most of it has to do with its ecology, with its invasiveness, and getting rid of it. And there's not a ton of literature on how to grow it for use in these facilities across the globe. Basically, everybody just uses the what we would call the invasive Phragmites. So as far as I know, this is really the first venture into evaluating the American haplotype group for its use in wastewater treatment. But I had to propagate it first. So I'm just going to sort of describe this process really quick. So I collected from these different counties, and these 3 letters are capitalized because that's how I sort of identified them from here on out. And I'm just going to show one population so you can get an idea of what I did for all of these populations. So let's look at Carver. So out of Carver County, out of this verified native population, I collected a bunch of rhizomes. That's my artistic, I have an art degree. Did y'all hear that? These are rhizomes. And I basically planted all of these rhizomes. And they did eventually get covered with soil, but this is my photo op with them to see what would grow and what wouldn't. And much to my delight, some of them did grow. And so I let them sort of grow out a little bit. And then to isolate these rhizomes, I transplanted these individuals into their own containers. And that way I was able to track the growth of individual rhizomes. They like to send shoots out, in some cases across those entire bins, leading me to misidentify which shoot was coming from which rhizome. So isolating them became very important. So let them grow out from there a little bit because I wanted to really leverage their ability to grow adventitiously from the culms to make a large number of replicates. So eventually I settled on populations that had a large number of shoots coming up, and I cut all those down, and I layered them in water so that would initiate that adventitious growth, and then crossed my fingers and hoped that this was going to work because the literature was a little bit sparse on this part of it, and I thought I had just cut down all of my research material. So, but fortunately, they grew a lot. They grew way more than I thought they were going to. And I had a great number of clones to select from, of genetically identical clones. And these were used as replicates in the following experiments. So I selected clones that were as close to as similar in size as possible from within each population. And those got transplanted and moved over to the growth chamber where I lived for the next few months. So most of my timeline thus far has been spent on propagating these things and finding a way to do it that really produced a large number of high quality plants for research. The actual experiment itself was just over 4 days. And on day 1 and day 2, I did leaf level gas exchange experiments. I was just looking at the leaf level. And then day 3 and 4, because this is really the important question, is how much water can it remove? I looked at the whole plant gravimetric measurements. And then there was one other variable I looked at, and that's VPD, which I alternated. And you're probably wondering, what is VPD? Well, you are in luck. VPD is the amount of moisture that the air can hold at a certain temperature, the difference between that and the amount of moisture that's in the air at that temperature. And it's temperature dependent. As temperature goes up, as the air gets hotter, it can hold more moisture. And this is what drives transpiration in plants. So as VPD increases, evaporative demand increases, and as evaporative demand increases, you very generally get an increase in transpiration from plants. But some populations it's not exactly linear for all plants. So the first thing I needed to do was sort of identify some of these populations that couldn't keep up at higher VPD. So like at low VPD, so here's two leaves, like we're looking at leaf level, low VPD, and they're both transpiring a little bit of water. And maybe one's transpiring a little bit more than the other, but it's not a lot. It may not, those differences may not really be detectable. But if we increase VPD, we really dry that air out, suddenly we start to amplify the differences between these two leaves. And as you see, this one's now transpiring much more than the other one. And so that's how I identified populations that I wanted and populations that I didn't. Is a very rough way of looking at it. So hypothesis one was that gas exchange traits like transpiration, stomatal conductance, which is transpiration and the flux of carbon and oxygen in and out of the plant itself, and photosynthetic rate, also referred to as carbon assimilation, will differ across these different populations. But I wanted to identify proxy traits too. Again, we don't want to do a common garden experiment every single time that we want to evaluate a population of native Phragmites. So looking at these leaves under a microscope is the best way to do that. So these gas exchange traits may hopefully correlate with leaf morphological characteristics like stomatal density and vascular architecture. So let's start with stomatal density. And basically, if I take these two populations, population one is the one that transpires more. Maybe that's because it just simply has more stomata. It has more openings to transpire from, maybe. But I also want to look inside of the leaf. So if we do like a leaf cross section, this is from a recent paper put out by a lab group. And it's one of the best photos of highlighting this. Is not necessarily what I'm investigating, but it's a good photo to show the differences in vascular architecture. So in the red, we have the metaxylem, which is the primary method of how water moves through this leaf. And we have the bundle sheath cells in yellow. And it becomes something of a plumbing problem. If you have a bigger pipe, then you can move more water through it. So there may be differences within the leaf, and maybe that will explain the movement of water through these leaves. That's the hope anyway. So I did these gas exchange measurements with an infrared gas analyzer that costs more than my entire education. And it measured transpiration, stomatal conductance, and then carbon assimilation. And it basically just measures, it analyzes the gas going into the leaf, and then it measures the gas coming out of the leaf. And then between those differences, you can basically infer these characteristics. So hypothesis two was that the whole plant water removal would differ among these populations, because again, ultimately, we're more interested in how much water these whole plants can remove and not necessarily what's going through their leaf in a 1 square centimeter area. And as a proxy trait, total leaf area may indicate maybe a reliable indicator of water removal per individual. So if we fill these 4 populations, these individuals from 4 populations up with water and give them some time, maybe we see differences in water removal, and maybe that correlates with leaf area. And that's basically what I did. Filled all of these up with water to field capacity, and sealed the pots up so that the only water loss was being done through above ground biomass, and measured that change over time. And at the conclusion of all these experiments, we defoliated all of my newfound plant friends, and we stripped all the leaves off, and we ran them through a leaf area meter to get an idea of what's the total area of leaf that we were looking at per population. And then I was able to average that out across to get how many milliliters were lost per square centimeter per whatever unit of time I wanted to look at. And then hypothesis 3, if we can throw it back to collection, is that the individuals collected from these robust populations that we sort of honed in on early would transpire more water than individuals from the typical weak growth, non-robust populations. So just very simply that those individuals from robust populations would transpire more. And I got some results. I will share some of these with you. So when I averaged in total leaf area, and these are the top row of bars is from the high VPD experiment. The bottom row of bars is from the low VPD experiment. These were one day after the other. When I averaged out water loss through leaf area, there were no statistically significant differences across any of these populations. The bottom left, the LES population with the 2 red asterisks is the invasive, the one that is known to dewater biosolids at an exceptional rate. And the one next to it with the blue asterisk is it was the weak population, which as we can see doesn't look so weak anymore. So when we removed that average, the leaf area average, there were differences in these populations. So I did ANOVAs within each treatment group, and then post hoc tests to get groupings. And as expected, the invasive used more water at low VPD, but the next closest population was the weak native. With those 2 results right there. And then this was repeated again at with the high VPD experiment. And once again, the invasive removed more water overall, which is to be expected. And again, the so-called weak native more or less kept up. So if they're not averaged out, if there aren't any differences, but if we look at the whole plant, there are differences. Why would that be? The simplest explanation so far is that the invasive just grows a heck of a lot more leaves than just about any of the natives. The next closest in the native genotypes was that weak control, almost. Other than that, there were no statistically significant differences in leaf area across any of these native populations. So that's about where I'm at with my project right now. The next steps for me are to advance the 2 controls and 2 native populations into another round of gravimetric measurements, except this time, instead of doing them on a small scale, I'm going to grow them out. This is my test run that you're looking at in these 80-liter bins, where they are humongous and extremely heavy. And I want to measure their water use very similarly to the past gravimetric experiment over their growing cycle, over the whole growing season. So I have a whole next round of plants being propagated right now. And I hope to do these measurements over the summer and into the fall. Whatever results I get from that will be distributed to stakeholders, in particular, the wastewater treatment facility operators. Again, it is their discretion what they want to do with these reed beds. But hopefully I will find something that I can share with them so that they can move away from using this invasive. And then if we identify a promising candidate within this experiment, a promising population that may be advanced on to doing in situ trials in a wastewater treatment facility that's been cleared out for our use. So I just want to thank my advisor, Waleed Sadak, who's also on my committee, along with Dan Larkin and Sue Gladowitz. I want to thank Julia Bonin, especially. She has been an incredible resource for me, both from collecting and propagating and just general care and just bouncing ideas off of her at every opportunity. Within my lab group is Daniel Monins and Chansu Ding, and then Janelle Balmaceda, who is another master's student in Dan Larkin's lab, working on Phragmites in another capacity. And then, of course, I want to thank Mazurk for funding my education. Thank you. That's all I got. I guess we're taking questions now. if you are in chat, it would be much easier for me to read your question than hear it. But of course, I leave that up to you. Yes.
Great job.
Thank you.
Yeah. So you talked about using the native plants in the wastewater treatments as like not being worth their time. And so I'm like wondering what that means, right? They just like I feel like that would be better than just leaving it open, right?
Sure.
Or like, even if they didn't want to use it, could they just like cut down those seed heads? Like I feel like there should be options.
Yes. So the mixed results, the question was about the native Phragmites that are currently being used in wastewater treatment in Minnesota and the sort of mixed results reported back about that. That is sort of secondhand information for me. And these operators weren't, I would not say they're not doing sort of like robust hypothesis testing on their, I think they just collected native populations and put them in there. What that means, I don't know. These things can take several years to establish. 5 years was what I was quoted from an operator, 5 years to establish a reed bed. So it may be that they are still establishing. It may be that they are established and just aren't removing water adequately. I don't think there's really a hard metric being adhered there. I think it's just sort of somebody's subjective. They're not doing very good. Yeah.
Thanks for a great presentation.
Thank you.
I had 2 questions. The first one is you mentioned about the rhizomes propagation. Do you know how do you perform? Like you just put the rhizomes in the soil and they grow?
Yes. So I pulled from a few different existing papers. Like I said, they were very vague in what they did to actually produce their research material. But one thing was clear, we just planted rhizomes in half sand and half peat. And so that's what I did. I just followed them. The general idea that I got from reading literature was that this is a plant that will grow basically anywhere and just about anything. And so I thought that while half sand, half peat isn't like the ideal substrate that I might use on anything else, follow the literature, do what the literature says, and it worked. I think they have a sort of pH preference that leans on the acidic side. And I think the peat acidified that a little bit and made propagation a little more successful, but that's just a guess on my part.
My second question.
Yeah.
Would be, I noticed that some photos, the Phragmites were very, very tall. Is there a record on how tall they were? You know.
I saw 15 feet quoted quite a bit, but there are a few pictures floating around out there that these things had to have been over 20 feet, but I don't know if that's just, a photographic effect or like I'm looking at a very small person standing in front of a Phragmites patch. The question was about how tall the Phragmites culms can get. Yeah, so. Okay, so I got a question from someone. So how did you verify the clones were genetically identical? Well, mostly because I got them off of the same source material. So anything growing from, so I have the culms, the stems, and I layered them in water, and then from each node I would get more growth. So I just very religiously tracked which culm was which. I tagged everything individually. Anytime that maybe a culm sort of came apart in the water and I wasn't able to positively identify the source plant, I just discarded it. And I had enough material that I didn't really see that as being a big deal. So basically, I just extensively tracked everything. I tagged everything, and I just kept very good accounting of where everything was and what source plant it came from. So I have a timeline all the way back from those initial rhizomes that I collected with unique identifiers and where they got planted in a bin, what grew from them, how many of those stems I harvested, how many replicates I got from those. I just was very thorough with it. Anyone else?
Next steps.
Next
What
step.
are you doing next?
What am I doing next? So I have a large, so I showed that picture of the, we'll show it again. Those large bins, those 80-liter bins. I have the 9 of those right now in the greenhouse, and they all pretty much look like that. I have a ton of material, source material to start with, way too much. So I'm hoping to very quickly identify the populations that I want to advance into this next set of measurements, so I can sort of clear out my space a little bit and get those layered. Basically at this stage, they're ready to be layered in water. And that process, when I did it sort of in cooler temperatures, took about a month and a half, but as it's warming up, I expect that it'll take a little less time than that. And I'm hoping to have a whole other set of research material individuals to start growing them out individually in about a month and a half.
Is there a way to sort of do some of the measurements that you're doing in the greenhouse here on your material from in the greenhouse and under these conditions? Can you like take that to the field, or is that like not an option? And I mean, obviously outside of the scope of this project, but like, are those technologies like transferable to?
Yeah, I'm going to repeat that question.
Okay.
Tell me if I got it right. So the question was, am I able to take the research material that's been in sort of controlled conditions, like the growth chamber and the greenhouse, and take that out to the field and replicate these experiments? I would say yes and no. So the benefit of the growth chamber is that you really minimize environmental variables. Like a phenotype is your genotype times the interaction between that and the environment. So if we minimize environmental noise, basically, then we can sort of isolate these phenotypes and say that, like, okay, we think that with identical conditions, we can say that this one transpires more water than this one. I don't know how well that would translate to a field experiment. I think, of course, you start losing your control over the system once it gets outside. So the thing is, I don't know. I've never done, this is my first time doing this indoors. And I imagine there are some precautions you could take outdoors or steps you could take to sort of minimize that noise. I don't know how well it works. So ultimately, I think once we get these things, because these reed beds are pretty big, once we get these things installed into a reed bed, the identifying these traits at like the individual level is no longer the question. Now we're looking at the entire stand and how much water can it remove across the entire stand. And so then, like, the question is, like, basically how often are these facility operators having to flood these beds and replace the water that's in there? So that would be my guess. Yeah. All right. Question: What do you think about using the native frag as the species for water reclamation, et cetera, instead of the invasive one? Wouldn't that be advantageous as a native? Yes. So, I mean, my personal opinion on this is that using the native, even if it does not perform as adequately as the invasive, is already a step up from using the invasive. And that's coming, I'm sort of, like, I would say, a self-described environmentalist, and I care about preserving native species, and I sort of care about preventing invasive species from taking over ecosystems. So in my opinion, it's absolutely worth using the native, even if there's a drop in water removal overall. Of course, not everybody sees it that way. Some people do look at it very simply as how much water is being removed. They don't care if it's native or invasive. They just want it to remove water. And that is their viewpoint and their opinion. So, we do have to sort of try to meet halfway on that one, which is why I'm trying to find a well-performing native. Oh, and so they asked what program did I run my stats on? R. I just used base R for my ANOVAs. And then I am using a mixed effects modeling package that I did not present today, using these reps as repeat measurements, and that is LME4. And that's something that I'm still working on. So I do not want to present that to you today. All right, you got anything else? Nope. All right. Well, thank you everybody who's watching, including it looks like my mom is here. Thanks, Mom. Thanks for watching. Appreciate it. I